See what the. I guess I can go. I think there was a whole, yeah, like all of us. Something soon.
Good.
Okay.
Okay. So I'm presenting on STING-mediated T-cell apoptosis. First, a little bit of background. STING is the central sensor of cytosolic DNA. So typically, it might be caused by virus, bacteria, or damaged self-DNA. So generally, it's a bad thing. So cell develops a mechanism to combat that. So when STING is activated, we can get enhanced antiviral defense and also cancer immunosurveillance. Here's the overall of the STING pathway. So first of all, we can see that the cytosolic microbial double-strand DNA activates the enzyme cGAS, and the cGAS catalyzes the cyclic GMP-AMP. And the cGAMP binds to STING in the ER. And STING recruits and activates the kinase TBK1, which phosphorylates IRF3. And the phospho-IRF3 moves to the nucleus, which induces type 1 interferon expression. So STING is classically studied in macrophages and dendritic cells, and it is the type of cells that we usually associated with DNA sensing. But a paper by Larkin et al. Showed something surprising. We see that T cells themselves carry a functional STING pathway, and when activated, it can trigger a full interferon response as well. So here's our first figure. It shows that T cell exhibits a functional interferon type 1 response to STING agonists. So STING can be activated by the synthetic small molecule DMXAA in murine cells. So in panel A, we can see that the murine cell robustly express STING protein at levels comparable to macrophages. So, oh, sorry, I'm sorry. So here's the Western blot. We can see that the bands for the wild-type T cells is somewhat near or even larger than the wild-type macrophages. It shows that the T cell is expressing STING normally. And here, gap pH is the positive control. And we can see that the negative control, which is the STING knockout, does not have the band. And when we look at the panel B, we can see after the treatment with the DMXAA, we see an increase in all of these ISG genes, which I will explain later, like increased like transcription of all of these genes when we activate STING pathway by DMXAA. And in panel C, we can see that STING promote robust expression on both IFN beta and IFN gamma. And I will just skip panel D. On panel E, it is showing that when the T cell is activated by anti-CD3 and CD28, there's an increase in the production of IFN, while the knockout of CD3 removes that effect. And on panel F, you can see that both CD4 and CD3 has the production of IFN beta, while it seems like only CD4 has the ability to produce IFN gamma. And on panel G, we can see that DMXA lights up all of these cascade of STING pathway, including the TBK1, the NF-kappa B, and MAPK signaling, while the STING knockout does not. So the overall takeaway message is that T cell exhibit a functional IFN1 response to STING agonist. So we see that upon activation, T cell has the ability to produce IFN beta, IFN gamma, and then also the ISG genes. So for the IFN beta, it is also type 1 interferon. It can induce ISG genes, and also it boosts antigen presentation. So it might increase the MHC class 1 on T cells or dendritic cells, and it can also enhance antiviral and anti-tumor immunity. And for the IFN gamma, which is also the type 2 interferon, it is also the hallmark cytokine for activated Th1 cells and CD8 T cells. It can also activate macrophages and support cytotoxic response against infected or tumor cells. And for the interferon stimulated gene, it is transcribed after type 1 or type 2 signaling, and it can enforce an antiviral inflammatory and can enforce the cell to be in an antiviral inflammatory state. So here's our first engagement question. If a T cell triggers STING pathway, unleashing IF and beta and ISGs, which means it is pro-inflammatory, would you expect that T cell to A, proliferate, B, no change, or enter a cell cycle arrest and apoptosis?
Human fetus in 20 weeks old. So you can see that the red ones are
Proliferate? Yeah.
red two, and the green ones
Yeah, so actually DMXA inhibit T cells proliferation when STING is intact. So it might sound like unnatural by sense, right? Because it's pro-inflammatory and it should be proliferating. So here's a CFSE diagram for the proliferation assay. We learned in class that the more dilution of the CFSE means more proliferation. And the left shift of the curve means the cell is dividing. And if the curve overlap with the gray area here, which is the onstin, it means it is not dividing. So here on the panel A, we can see for the wild type mice, anti-CD3 and CD28 has the ability to proliferate normally. You can see the only line here, the only curve on the left is the blue curve, right? So interestingly, when we add DMXA addition to that, the curve does not move left, means the proliferation is inhibited. And when we see for the STING knockout, when we don't have STING, the inhibitory effect was removed because the red curve now aligned with the blue curve, meaning the DMXA now does not have an effect on the proliferation inhibition. And for the third figure here, which is the inhibition, which is the knockout of IF1, we can see that it's a similar figure with the wild type. So it actually shows that the STING inhibition does not require type 1 interferon. And on panel B, we can see here's the timing of STING. So we have green curve, orange curve, and red curve representing we add DMXA on day 1, day 0, day 1, or day 2. And we can see that on day 0, we have a strong inhibition of proliferation, while on day 1, we only see a partial inhibition. And on day 2, we only have a minimal or no inhibition at all. So the conclusion here is that STING must be activated early, or at least in the first 24 hours to show like an inhibition effect. And the overall takeaway for both panel A and B is that DMXA inhibits T cell proliferation when STING is intact, and that is not by interferon, type 1 interferon. So here's our second question. When STING is strongly activated in T cell, what usually happens?
Yep. So let's do a prediction exercise. So if FETH2 controls neural TRAs, so that's tissue restricted antigens, what autoimmunity phenotype do you expect in FETH2? So
Yeah. Okay. So here's the mechanism of STING-mediated T cell apoptosis. So also a little bit of background. AIFM1 is a critical factor for STING-induced apoptosis. So it is short for apoptosis-inducing factor mitochondria-associated 1. And in healthy cells, it helps maintain mitochondria structure. But in apoptotic cells, it translocates to the nucleus and induces DNA fragmentations. So it is upregulated upon cGAMP and stimulated in T cells. And STING also induced chronic activation, ER stress, and unfolded protein response, leading to T cell death. So this is the figure showing that AIFM1 is upregulated upon the cGAMP, which is the protein we see in the STING cascade stimulation in T cells. So when we look at panel C here, the IAFM1 mRNA increases as time goes when we add cGAMP, right? So we can see the mRNA transcription peaks at about 9 hours, indicating new transcription in response to STING. And in panel D, here's the protein level of AIFM1, showing the similar pattern with the mRNA that you can see the bands grows larger and larger as time goes upon the cGAMP activation, confirms that the translation follows the mRNA induction. And in panel F, this is the apoptosis assay. We can see that cGAMP induces nearly 45% of the apoptosis in wild-type T cells. And when we knock out the AIFM1 gene using CRISPR, we see it greatly reduces apoptosis cells percentage. So the takeaway message here is that AIFM1 is essential for STING-mediated T cell killing, and its expression is increased upon cGAMP. So our third question is, if you block AIFM1 in STING-activated T cells, what result do you expect? Is it more cell death, or less apoptosis, or higher TGF-beta expression?
And this is a negative.
Yeah, you will see less apoptosis. So here's my exam style questions. So what conclusion best fits the data in panel A? DMXA increased T cell proliferation in all genotypes. B, DMXA suppressed proliferation only when STING is present. And C, IFN-AR deficiency enhances DMXA-mediated inhibition. And D, STING knockout T cell fail to proliferate under any condition.
Another question. If red signaling is blocked, what would you expect? The first choice is clear decrease on. Second choice is SRF2 decrease on. The third choice is both decrease. And four is no decrease.
And then the last figure is gonna talk about supposed to be meant for osteoporosis. It does.
This might be, so would you say your presentation is done for
It's from a
section?
Oh, yeah, this is all exam style, but
Given our timing, we might have to just send everyone's presentations and exam questions through our email chain.
okay, yeah. So yeah, that's all for my presentation. Thank you all.
Then,